What is wrong with this blood sample? Let's finally go back to top hat. Yeah, so this blood sample has too many. But yeah, what did we see? So in the previous one, we saw, yeah, we see the normal sample on the left, and this one has a lot of these purple spots. So these look more like white blood cells, right? And so, yeah, a lot of you said white blood cells are leukocytes, and those people are correct. There are way too many of these white blood cells. And this is a condition, and I know we might run over time, so if you have to go to another class, by all means, go on. This will be up. Yeah, so that's a lot, and that's an extreme example. This is what we generally call leukemia, and leukemia is actually a category of diseases. So there are four main types of leukemia, and there's also other types, but the four main type, well, they can be categorized based on like how fast did the population of white blood cells increase. Was it gradually over time, or did you all of a sudden have a big spike and the white blood cell population exploded? So acute would be that sudden spike in the increase of the white blood cell count, whereas chronic would be kind of elevated over time. It also depends on the maturity and characteristics of white blood cells. So what do I mean by that? Well, there's two main subcategories. It's called myelogenous, or sometimes called myeloid, or lymphocytic or lymphoblastic. And that might sound familiar, right? So it depends of, okay, what's the time span that these white blood cells increased? Also, where were they derived from? And this is why it's kind of, especially when you're studying leukemia, that it's important to know myeloid versus lymphoid. So it's like, okay, these cancerous white blood cells come from this branch of the family tree, or did it come from this branch of the family tree? So that's why you have acute myelogenous leukemia. You can also have chronic myelogenous leukemia. You can also have acute lymphocytic leukemia, and you can also have chronic lymphocytic leukemia. So this is what you do. You take either one of these, and then either one of these, and you have the four main types of leukemia. There are other types, but again, this is how we name those four types based on the time span and their lineage. So if you have high white blood cell count, do you have leukemia? And you probably don't. The most common cause of a high white blood cell count, remember, your white blood cells their normal function is to be your immune defense, right? So you probably have an infection if you have an elevated white blood cell count. Like when I had appendicitis, my white blood cell count, I forgot, I wonder if I still have that paper somewhere, but it was like super high and elevated. I'm like, okay, something's definitely wrong there. But yeah, so again, but leukemia is that when you have that cancerous leukemia, you have these white blood cell like looking cells that take over that blood smear. So like in that extreme example we saw earlier. So hemophilia, it's actually kind of funny because again, like if you say you're like when we talk about something being hydrophilic, we say it likes water. So it's like hemophilia, you like blood? Are you a vampire? It's kind of an interesting like etymology, but it's like, so hemophilia is actually a group. So just like leukemia isn't just a singular disease, hemophilia, it's a group of bleeding disorders. So all these people are prone to excessive hemorrhaging and inability to form clots. So even getting something small like a small paper cut can result in uncontrolled bleeding. They can't stop. Like they can put all the pressure they want, and they can try to get all the gauze in the world, but that's not going to prevent them. They're still going to bleed from that small cut. So yeah, I'm actually censoring this because I don't know if that would violate YouTube or Twitch, but yeah. But yeah, this is what we have. So there's different types. Again, it's a group of hereditary bleeding disorders. And it's interesting. Sex linkage, I'll save that for whoever's teaching genetics to you if you're a bio major, or I think if you're taking, I wonder if they cover in pathophys. But yeah, men are more risk of hemophilia type A and B because some of these clotting factors are encoded on the X chromosome. Yeah, so but I think we don't really have time to cover genetics right now. So, why are there different types of hemophilia? So, hemophilia all involves clotting and coagulation. So, type A affects clotting factor 8, type B affects 9, type C affects 11. So, they all affect the coagulation cascade. The different types depend on which factor is missing. So, missing or non-functional in that individual. So people with type A hemophilia have problems with factor 8. People with type B have problems with factor 9. People with type C have problems with factor 11. Yeah, so hemophilia, it kind of causes anemia. So again, anemia is that lack of red blood cells and iron. So if this person is bleeding out and you're just like infusing saline into them, and not replacing the red blood cells and iron, then that could lead to anemia, right? So yeah, if these people aren't making these factors you need to make factor 10 to cause the clotting cascade, they're going to have a hard time forming a clot. So yeah, these people, this is why they can bleed out from a very small clot, because of a deficiency in this one protein factor. Now, how do you treat hemophilia? So hemophilia is that you can give them a recombinant clotting factor. So what do I mean by recombinant? So what you do is basically take the gene that they're missing, and then put it into bacteria engineered to make proteins. So these bacteria, they read the instructions on how to make the clotting factor, and they just crank out the protein of the clotting factor. And if it's the same sequence, it's going to be the same protein. It doesn't matter that it was made in a bacteria. If the amino acid sequence is the same, the protein polypeptide sequence is going to be the same. And they don't just take the bacteria and inject it into the person. What they do is they have the bacteria crank out a bunch of this protein, and they extract it, refine it, purify it, and then make it safe to give back to that person. Hey, that sounds a lot like recombinant insulin and recombinant growth hormone, right? So this is why genetic engineering is very powerful, because the people who have a deficiency in a protein, well, now we can grow it in a lab. We can make purified extracts of this protein so that these people can make up for the deficiency. So yeah, this is why people who have hemophilia, they can have these recombinant vials of the factor they're missing, in case they do end up having so it is biotechnology, and a very useful and life-saving use of this biotechnology.